Physiologic and pathogenic release of calcium through the ryanodine 1 receptor. We present three scenarios. 1. Normal muscle contraction and relaxation. 2. The effects of succinylcholine on muscle contraction in patients with a pathogenic RYR1 variation. 3. The effects of succinylcholine on muscle contraction in patients with butyrylcholine esterase deficiency and a pathogenic RYR1 variation. These animations describe general concepts about the physiologic and pathogenic mechanisms of muscle contraction. It does not describe the intricate details involved in these mechanisms. Scenario 1. Action potential during normal skeletal muscle contraction and relaxation. A neural stimulus triggers the opening of voltage-gated sodium channels, allowing influx of sodium into the cytosol, which initiates depolarization. Repolarization starts with the closure of voltage-gated sodium channels and activation of voltage-gated potassium channels, which allows potassium efflux from the cytosol. Scenario 1. Cellular events during physiologic muscle contraction and relaxation. The main cellular components of the neuromuscular junction are shown. In response to a neuronal signal, a presynaptic motor neuron releases acetylcholine into the synaptic cleft. Acetylcholine binds to the nicotinic acetylcholine receptor and opens voltage-gated sodium channels, allowing influx of sodium molecules into the cytosol. This initiates depolarization of the muscle cell, shown here by the blue wave. This depolarization event travels through the T-tubule membrane and induces a conformational change in the dihydropyridine receptor, which allows it to mechanically interact with the ryanodine 1 receptor. This, in turn, permits a calcium release channel to open and release calcium from the sarcoplasmic reticulum into the cytosol, which induces muscle contraction. Within milliseconds, the enzyme acetylcholine esterase degrades acetylcholine. Repolarization of the muscle cell is initiated through activation of voltage-gated potassium channels, which cause potassium to leave the cytosol. This results in muscle relaxation. At this point, the ryanodine 1 receptor channel is closed, and cytosolic calcium is pumped back into the sarcoplasmic reticulum via the calcium ATPase pump.